Hello, friends. Welcome to the second edition of Elets Diagnostics Leadership Summit. In this session, we shall discuss how the diagnostic segment is gearing up for the possible third wave of the COVID-19 pandemic. Joining us in this panel discussion are Dr. Sanjay Arora, MD and founder, Suburban Diagnostics. Next is Dr. Colonel Prabal Deb, Head of Operations and Director, Reference Lab at ESTA Labs. Dr. Deb will also be moderating this panel discussion. Next, please welcome Dr. Colonel Jyoti Kotwal, Senior Consultant and Head of Department of Hematology at Sir Ganga Ram Hospital. Please join your hands to welcome Dr. Partha Roy, Head Operations and Quality at Spice Health. And we also have the privilege of the presence of Dr. Disha Bhatia. She is National Head Microbiology and Serology at Code Diagnostics. Welcome, ladies and gentlemen, to this very special edition of Elets Diagnostics Leadership Summit. I would now like to invite Dr. Prabal Deb to moderate this panel discussion. Over to you, Dr. Deb.
Thank you so much. At the very onset of this August gathering, my sincere thanks to Elets for coordinating this galaxy of eminent scientists who have been engaged in laboratory diagnosis in the world of COVID. I think today we are touching about 1.5 years of the onset of COVID operations in India, and it has given us set a lot of milestones. It has redefined lives. It has made our understanding of COVID, of immunology, and various challenges related to diagnostic segment in a totally different way. When we talk about COVID, what we have learned in the first and second wave is very important to set the pitch for the third wave. The eternal question everybody keeps on asking, is there a third wave? When will that happen? And how are we going to combat it? The first two waves has let us learn many things about how to scale up our operations, how not to be taken by surprise. How about the supply chain management to ensure that whatever mistakes we made in the first wave, we thought in the second wave we won't be having that. But whatsoever was the reason, second wave also caught us unaware. And it is expected that the third wave is supposed to be of a higher magnitude. So, if we start off with our experiences during the first two waves and our learning thereof, if I can request Dr. Sanjay Arora to just share the learnings as to how his organization went about it and how does he recommend that we prepare ourselves for the third wave.
Uh, Thank you, Dr. Dev, for initiating this. Um, as you said very correctly, that we thought that from the first wave to the second wave, we would have learned something, but actually we were again caught by surprise. In our experience, we started doing few hundred tests to few thousand tests in the first wave, and we peaked to maybe around 5,000 tests a day. And we thought at that time that the agility of supply chain, as you mentioned, of being able to initially procure PPEs, collection swabs, VTMs, even kits was a big challenge at that time. To a large extent, that was managed fairly easier in the second wave, but I think the sheer volume of requests, outstripped the capability of the lab to manage that demand. So from 5,000 test requests a day, we went to almost 13, 14,000 test requests a day. And I don't think any lab, is able to cope up with more than 2.5 times the volume that it can manage. So what we actually did, and that's why I'm sitting in a makeshift office, is that we actually broke down our corporate office. Our molecular lab was on the floor above. So we extended our molecular lab, we extended it into our corporate office. So today we don't have a corporate office, and we continue to run the molecular lab from a larger facility where we have the capability of being able to test close to 15,000 tests a day. But as we ramped up that capacity, the demand came down. So it's always been, this sort of chicken and egg situation.
Actually,
Today, the demand has probably come down to, for us, maybe about 3 to 4,000 tests a day. So in our case, we have seen what we call as a very fat tail to even the second wave. It's not really come down to few hundred tests, which we were seeing in most other places. In Mumbai, at least, we are still seeing a decent demand of COVID testing, and now with travel being opened, the demand for POCT testing also has gone up. So I think the biggest learning is that we need to be prepared for, as you said, perhaps a higher volume, and that's why we've continued despite the volumes going down from 12, 13,000 to 3,000. We've still kept our capacity of close to 15,000 tests a day active. That our infrastructure is there. We've had to, of course, ramp down our manpower. So I think the flexibility of being able to rapidly ramp up manpower and being able to ramp it down, the visibility of how the numbers will, so your supply chain is able to cope up with the number of kits that you need. I think that agility is perhaps the biggest learning that we had from the first and second wave, and we're trying to keep that agility alive, keeping us on our toes even today.
thank you, sir. I think that is very pertinent what you mentioned that scaling up and scaling down and looking at the manpower because COVID operations is quite manpower intensive. Yes, the data entry operations has been automated to a great extent in the past, I would say, 10 to 12 months. But still, there's a huge manpower right from collection, the phlebotomist, to getting the logistics, coming into processing, and meeting the TAT of 24 hours. I'm sure in all states that 24 hours, and especially we have seen how in Mumbai they used to monitor on a day-to-day basis and get give
Yes.
warnings all around. So it was a very important thing that we met all these timelines. And how we also do it at our setup is On a day-to-day basis, we have a regular review about the situation, on-ground situation, since we have our operations in other parts, multiple states in South India. So we have our review, and I'm sure all the labs keep on reviewing the situation because, again, we expected the third wave to come sometime in August, then September. Now it is presumed in October or November. But you have to keep all your kits ready, and kits have got a definite shelf life, and their availability suddenly goes down whenever you are caught by surprise. So these are very important things, that the ability to ramp up, ramp down, the agility about the supply chain, everything is very important. So at this stage, we find that a lot many improved technology has come in because of the travel regulations. Like in the Middle East and other countries, they say 72 hours prior to travel, there's a RT-PCR, and there's a rapid RT-PCR 4 to 6 hours prior to boarding. So this has seen the onset of a lot of novel technology. Everybody wants to give a report faster for that. So, Dr. Partho, if I can request you to touch upon all the rapid and novel technologies which have come up, especially for travelers and rapid screening, please.
Thank you, Dr. Prabal. Yes, uh, as we have been actually seeing that in this last 1.5 years, the way the requirement of testing has changed, and I think for the first time, the research lab has come into the clinical laboratory for the benefit of rapid testing, surveillance, and maybe even seeing where the mutations, etc., are going. Now, having said that, uh, countries worldwide have changed their strategies of testing. So, initially, of course, the most important thing was may not have been rapidity, but very good sensitivity and very good specificity, like RT-PCRs. Then we had some amount of rapid testing, like rapid antigen testing. And then we came out with the antibody testing, which initially was mostly because of for serosurveillances and not too much for diagnostic purposes. Now, yes, the same technologies that we were the gold standards had, everybody had, each of them had certain drawbacks. For example, RT-PCR had this problem of multiple steps. Like you have an extraction step, and you have got the master mixes and so on and so forth. So it really could not be done at a POCT level. So I guess that this was really a requirement of that time, and now we are seeing the fructification of that. So overall, as Dr. Prabal has rightly said, that travel has become important, but people are also really worried that what is happening between that 3 days prior to the RT-PCR and as you are boarding the aircraft, have you got something or not? So the same technologies that were being used are now being transferred and modified to go into a kind of a POCT model. And that is also helping us in the way that not only it is rapid, it's also fairly sensitive because these are all molecular assays. And the novel technologies associated with this, whether it's an RT lamp or it's RT-PCR or it's a CRISPR going on to an RT lamp or RAA and things like that, have retained the sensitivity. It's retained the levels of the level of detection. So when we talk about the RT-PCR per se, now we do have in India at least platforms like Acula, Thermo Fisher, which works on a cartridge-based system. Now the entire nanotechnology or miniaturization is working in a way that the entire test is done. You don't have a step of any RNA extraction, so that makes the entire system fairly safe and rapid. And once you take the sample, you just put it in a kind of a cartridge, and the machine itself takes you through the various stages. So it can be used by a person not really very well aware of various pipetting techniques, etc. So that's one of them. The other platforms that we are seeing is something like an Abbott ID, which is of course a isothermal based assay. In this, the most interesting thing is that you can do 4 tests in an hour, and if something is positive, you get a positive report within exactly 5 minutes. So many times what's happening is that various private laboratories are offering this test. These are slightly expensive, but what you have is that you immediately get a result within 5 to 10 minutes, and maximum time it takes is about 13 minutes or so. So that is available with you. So it's a la carte menu. The uh, technology that is coming up very well is the lamp technology. Is isothermal based technology. Advantage of this is that you totally do away with your thermocycler, which was we had ramping up of temperatures going down, and it was a very complicated but very interesting technology. But lamp has brought in a very simple system in which these things are not happening. And though initially there were some uh, doubts raised that whether how sensitive it is, but it is definitely coming out to be a very sensitive, specific molecular assay. Also, it's coming out to be quite cheap. So that is a very interesting and important thing. Multiple tests can be done. There are platforms like the Mispalium or the Agapes, where you can do almost 32 tests in one go. So and it is accepted in the Gulf countries. Many of them are FDA approved, some of them are CE approved, but they are accepting that. So that's one more thing. In the
Yeah,
entire process, what happens is the rapidity part is so important that the rest of your process and procedures has to be very, very robust, so that you can give 200 reports maybe within a period of 3 hours. So that is the
that
important
is
thing.
very
That's what.
nice. That is very nice. Yes, I mean I do agree that rapidity today is ruling the rules, because there is a need for travelers and there are certain guidelines, and people also want to have the report fast. Yes, we are doing with the CRISPR technology at the airport operations at Bangalore. So multiple things are there, and everybody is looking for how soon can the reports be released. Yes, but what is happening now with all these testing being done, large number of testing being done, and especially during the first two waves, we've seen that a lot of high volumes were there. So, Dr. Disha, if you can just, give your thoughts about how were the quality control being maintained with such large number, which Dr. Sanjay had mentioned that yes, about 5,000, tests labs were doing. So that means quality maintaining that it's a major challenge. So what are your thoughts and how were you dealing with that?
Definitely, Dr. Prabal, this was a major concern because suddenly we were hit with a demand of thousands of tests per day, and the second wave was just unrelenting, and a lot of kits were there in the market. So there was also this pressure that we have to be ready with kits, as Dr. Sanjay mentioned. So first and foremost was the need to validate a number of kits, because even though kits were being ICMR approved, we experienced that ICMR would approve them for a particular lot, but then robustness checking was up to us. So because lot to lot there would be variations in the kits, and everyone was learning on the go. So with a new batch of the kit, they would change the interpretation, they would change the CT cutoffs, and even things like what are inconclusive results, when do you repeat a test, or when do you ask for a new sample, or if one gene target is positive, is this positive, if both the gene targets are positive, is only that positive. So these things were constantly changing. So we realized that we needed a protocol which like we have for all our other tests, But even in this situation of so much burden of tests per day, it was very important to stick to our protocols of validating each test and maintaining our internal quality assurance protocols in addition to our external quality assurance like CAP and NABL and the surveys they send and the EQA samples. So it became very important to do that, and also to train and have regular training for all the staff involved. So
Very
as Dr.
important.
Sanjay mentioned
Very
that
important because you already have a change of manpower, everybody's frequent movement. So training and ramping up your resources very important to train them. I do agree.
yes. Yes, so repeatedly you would suddenly have new people and they suddenly have to jump in and do non-stop testing through the day. So, also taking into account that there are so many shifts and they are tired, so it became very important to keep stressing
Mhm.
on quality control and also on infection control and biomedical waste management. So, these became really important.
But do you think that so many testing is being asked for? Is it like for whether the rapid antigen test or the PCR? Okay, is it actually required, or there are too many tests being ordered, which is putting a burden on the labs, especially during times of crisis when the actual patient tends to get delayed. The reporting of those samples gets delayed. Like corporates opening office, in that resources are required. So, what are your thoughts on that?
Definitely, Dr. Prabal. So in April and May, the situation, like I'm talking about Delhi NCR, the situation was really bad that, there were patients who were waiting that they were unable to start with their management or with an operation without a positive report. And the labs were just burdened. People were getting dates
Yeah,
for, 3 days later for testing. So what we realized was that there were people who were giving multiple samples per patient. People were also sending samples to multiple labs at the same time and repeating samples at a gap of 2 3 days, wanting to check their CT value trends.
exactly.
So, and then they would compare that, okay, this lab gave me this CT value, you gave me that, and 2 days back my CT value was, higher, now it is lower. What does this mean? Is my illness getting worse? So that's when we decided we were getting a lot of queries also regarding this. So we decided to make an information booklet which we were sending with each report, which was like a guide to understand what all tests are there and what each test means, and explaining that the CT value is not an indicator of your infectiousness or of your prognosis or even of how long you have to self isolate. So these became very important, and
Yeah,
to
I,
tell
I
them.
agree. I agree. These become very challenging and puts a lot of burden on the labs. And especially for the lab, it is just not doing the RT-PCR or the rapid antigen. There are other things like in the month of news reports which came up in month of May last week that in a span of 5 days there were 179 cases of MISC, that is a multisystem inflammatory syndrome in children alone in Delhi NCR. So that is something which becomes a concern, whereby there are children, and now it is well known in adults, and in adults who are exposed, or children exposed to any COVID or had a COVID bout before, or even after immunization, this multi-system inflammatory syndrome is being reported. So for workup of that, it requires a lot of lab support, and if the lab is burdened, it can cause a lot of challenge in reporting. Like if I can understand from Dr. Jyoti regarding the D-dimers. D-dimer is such an important test by which patient treatment and prognosis outcome is so important. So lot of challenges in D-dimer test. If you can just enumerate your experience, please.
Yeah, D-dimer becomes a very important test in deciding whether the patient has to be put on high dose unfractionated heparin or a low molecular weight heparin if he is a severe case, or if it's a mild moderate. And we saw it in the second phase that they were put at, most of the patients had to be treated at home in Delhi, and they were put on DOACs going on the basis of the D-dimer. Now, at this juncture, I would just like to talk about the issue of a false positive D-dimer coming because of heterophil antibodies in patients of COVID. And we picked up this case way back in the first year only itself, when there was a very senior doctor in one of the government medical colleges who had COVID, had D-dimer, was fine. And when he was asymptomatic, they did his D-dimer and it was very high. Now, that hospital was using a photo optic coagulometer. So they sent the same sample to another hospital, BLK, and to a private lab. Now, the private lab gave a negative report, less than zero point 0.5, but the other hospital again gave a report 2000 FEUs. So that's when the sample came to me. Now, we shifted, though we have two photo optic coagulometers for years, we do all our coagulation on that. But 3 years back, we shifted only for D-dimer to an electromechanical, knowing the issues of D-dimer in electromechanical, especially on the patients who are serious, who are on ECMO, who have hemolysis, or who have jaundice. And we do D-dimers mainly in ICU patients. And lo and behold, we did it on the photoelectric. It was high, and it was absolutely normal on the electromechanical. So this issue, diagnostic people and all of us need to understand, because hematology, we tend to not go into the depth of it, that you may have a false positive D-dimer, so it has to be clinically correlated. In fact, if one goes back, there was an article published in 2003 in the British Journal of Hematology about heterophil antibodies causing false positive D-dimer on photo optical. So the Vidas is not going to give that. The Stago is not going to give that. But the others may give in almost 5 to 10% cases. And following this in our patients, we ran a parallel on both the equipment just to see how many we get. You may get it in post-COVID patients who are on follow-up, almost 5 to 10%, you may get a false positive D-dimer on the photo-optical one. So this issue of what equipment you're using and that you may get a false positive, which may
Yes,
not clinically
yes.
correlate, is a very, very important thing that we have to keep in mind for the third wave, rather than just labeling them, because we've had patients who've been given longer duration of anti-oral DOACs because of that. They've been given antibiotics which interact with the DOACs sometimes, and then we've had cases coming with bleeding too. So it's very, very important
Agreed.
to understand
Agreed.
this issue of
Agreed.
D-dimer.
No, in fact, if we can say that, I mean, what was initially thought about, that COVID is a respiratory illness. Okay, rather than that, we can see that it is more of a hematological, a multisystem manifestation of a hematological illness. So the hematology workup is extremely important, and for that, the mainstay remains a D-dimer, a correct D-dimer result. Beyond that, we have seen a lot of importance about HIT, a lot of reports coming about HIT in COVID. And what are your thoughts and anything about the PF4 antibodies? If that has been a concern in second wave, but very poorly understood. And I would presume that in third wave also it would again take a main stage. So, what are your thoughts on that?
Yeah, at first, I would just like to say that heparin induced thrombocytopenia can be a fatal thromboembolic phenomenon, which is drug induced because of antibodies which develop against the combination of platelet factor 4 and heparin. And severe patients of severe COVID-19 patients do get low molecular weight heparin or unfractionated heparin, especially when they go on to the ECMO. And that is the time when they are likely to develop HIT. Now, there's data from the French ICUs where they have found that normal patients who go into ECMO or into ICU, the incidence of HIT is only 2%. But in COVID-19, they have reported it as 21% from the multiple ICUs of France. So that brings into the issue that it is a common problem, and specifically in patients on ECMO where they are given unfractionated heparin. And the dosage of heparin has been for quite some time. First, they were on low molecular weight, became worse and went into unfractionated heparin. So comes in the role of anti-PH4 antibodies. Now, for HIT, the anti-PH4 antibody titer should be more than 2 OD. So you can use the rapid test for the diagnosis. You can use the automated Liam, which is available, and it's being supplied by the IL and Werfen people, and people are using that. One can use the gel cards which come from the Biorad, which is for anti-HPF or IgG antibody. Very good test, which comes out within 10 to 15 minutes. You have the report ready, or there are these cards available from Stago Diagnostics. Now, or you can go in for the ELISA, which has its own issues. It's also available on the automated coaglometers, but it's got a very short life for 100 tests, and one, it's not cost effective to be used. So coming to that, anyone who's getting heparin can have anti-HPF4 antibodies without having HIT. So it's very, very important, because a person who's on with COVID itself is a thromboembolic disease. It will have slightly reduced platelets. It will have problems of thrombosis. It will have neutrophilia just like it. So it becomes a little difficult to differentiate between the two. So the 40 score, which is a clinical score of the temporal relationship of the thrombocytopenia, the degree of fall of the platelets, and thrombosis has to be kept in mind, and as well as any other cause of thrombosis. So if the fibrinogen is low, then it may be DIC, which is more important rather than HIT. But at this juncture, if the anti-HPF4 antibody by any methodology is positive, patient should be considered as probable HIT. And the confirmatory test, this is the antigenic test, is the functional assay. Now, the serotonin release assay, which is done with the radioactive material, is not available in India. Abroad also, in most places, it's not available. So the other test is to look at heparin-induced platelet aggregation. As of now, in the whole country, I think we are the only people doing it for the last 10 years. First, I was doing in the army, and now in Gangaram, where we look, patient serum is used to activate normal donor platelets in the presence of various doses of heparin. And in COVID, we have seen in two cases of ours from the ECMO that there was, who had sepsis, there was spontaneous HIT, which is an autoimmune HIT, which can occur even in the absence of heparin. So there it's heparin independent platelet aggregation of donor platelets by the patient's serum. So this is the gold standard diagnostic test, not only for HIT, but even for VITT. So in VITT, you need the high titer anti-HPF4, but not for HIT. So you can do it with these rapid diagnostic tests and then go for a confirmatory if possible.
Yeah.
It's
Yeah.
one thing I would like to tell, there have been cases we're getting from Kerala now, from KIMS, where patients had VITT or HIT, but the test came negative from labs doing it on the LIA system, is because the samples need to be serum has to be complement inactivated within 2 hours before transportation. So I would request all the labs who are listening to this, because their cases have come false negative, a number of them, please inactivate the serum for complement before transporting it to your parent laboratory. Otherwise, you'll get a false negative anti-HPF4 antibody in a patient who's positive.
I think this is a very important message to everyone, especially since in COVID, the time window available is very short. There is no chance of again getting a repeat sample, because whatever action has to be taken has to be done in a very short span of time. So thank you for all these updates, Dr. Jyoti. And to move ahead, if I can request Dr. Sanjay, on your thoughts regarding the various available antibody tests that are there, and is it actually a reliable measure of the immune system, and how does it predict need for boosters, which has been under discussion right now? So, Dr. Sanjay and Dr. Disha, I'll just request you both to come in one after the other and share your thoughts on this, please.
Sure, sure. So I think just as Dr. Disha mentioned that there's a large number of kits that were available for RT-PCR. I think probably an equal or if not larger number of antibody kits became available. Total antibodies, then IgG, IgM. So every company had, so many different types of antibodies, and as Dr. Disha said that validation of the kits became a huge challenge, and if you are a large lab, then you need to test so many different samples on so many different kits. What we have finally narrowed down is basically 2 antibody tests that we do. One is the nucleocapsid antibody test, which we find is a good response post a natural COVID infection. And then, of course, is the spike protein antibody, which is a quantitative assay, which we do in response to vaccination. I think it's very important for both doctors and patients to be aware that when they prescribe an antibody test, they need to be very specific about which antibody test they should ask. Because many times, if you ask just an antibody test and if you do the nucleocapsid antibody, that's not going to show a response if it is to be done post vaccination. So it's very important that somebody who has taken vaccination but gets the wrong antibody test done, it can have a misleading interpretation that, oh, my response to the vaccination was inaccurate or I'm a non-responder. So for
But one
natural
basic
infections,
question, basic doubt here, sir, sorry for interrupting, that
please.
there are many guidelines which lay a question mark on doing antibody test post vaccination. There are FDA guidelines also or advisories against it.
Absolutely.
So, what
Absolutely.
are your thoughts on that?
No, no, I think as per guidelines, I think the guidelines also unfortunately continue to keep evolving. WHO, CDC, all government guidelines continue to keep evolving. I think it's also important as lab professionals, we also need to sort of internalize and think what is relevant and what is not. At this point of time, unfortunately, the only measure for a doctor or a patient to see if there is a response to vaccination is to see an antibody titer. There is no other way for them. And because of the anxiety, because of the uncertainty, I think this is an intermediate tool, I would say, to at least allay any doubts that am I a vaccine responder or not. Uh, as per your question related to whether this is a measure of immunity, uh, I can give you a few examples. We've had some of our internal staff members who, after a natural infection, continue to show presence of antibody for almost 12 months after their natural infection. Whereas when we see post-vaccination, we see the vaccine titers dwindling, say 6 months after the second dose. In my case, particularly, my vaccine titer was maybe in thousands, and I did mine just few days ago, and it was 600. So uh, this is, of course, one of the measures, but we can't measure the T cell immune response. That's still not easily available to everybody. So it's still a combination of both the B cell and the T cell response that we need to see. But as you said, I think these are still steps that we are taking, which are intermediary, which are just, we are sort of on the fly. We are trying to adapt and adopt, finding some way of giving some comfort to people. Yes, are you a responder? Are you not a responder? And is
Yeah.
your titer going up or going down?
It's more in
But over to
evolution.
Dr.
All the
More in
thought
evolution.
process more in evolution right now. Yes, Dr.
Yes,
Disha, what
yeah,
are
yeah.
your thoughts on this?
Yes, so very true. That distinction is extremely important. That is the test targeting the nucleocapsid protein or the spike protein. In addition, there are many assays which are qualitative or semi-quantitative. So while they will generate a number on the report, that cannot be treated as a level. So that
Exactly.
awareness needs to be there in clinicians and patients that
Very
this is not a
pertinent. Very
quantitative
pertinent.
result. And also there are two types of assays. One are which are binding antibody assays, and the other are neutralization assays. So most of the assays that are available are binding antibody assays, which cannot give much of an idea about the protective effect of a vaccine or a natural infection. These are just going to indicate the presence of a past or a recent infection according to your IgG or IgM levels, the exposure status. Whereas the neutralization assays are the truest reflection of your ability to fight off an infection. So there are three kinds which are available. The best kind, which is used in research, is the neutralization assay, which are the PRNT, but these require BSL-3 labs. So these are not commonly available to patients in diagnostic setups. Then there are pseudovirus neutralization assays. These are also not commonly available. So there is one test which got FDA approval, which is the CPAS by GenScript, which is a competitive ELISA, which tests this neutralization, mimics the neutralization reaction in vitro. And this is actually the closest we can get to measuring the actual blocking effect of the antibodies towards the entry of the virus into the cell. So I think we should be very clear what kind of assay we are opting for, what gene it is targeting, what protein it is targeting, sorry, and whether it is actually quantitative in nature. And also it is important to remember that, we don't have one level above which we know that we'll be protected or below which we won't be protected. We have not yet established, even for quantitative tests, that what is this magic number of neutralizing antibodies which are going to protect us against an infection. So there is,
Yeah.
no clear answer yet.
And overall, I would say a lot of importance is given to antibody testing only. And in the passing, which Dr. Sanjay also mentioned about the T cell immunity, many a times we are overlooking this T cell arm of the immune system. And Dr. Jyoti, if I can ask you, what is the role of T cell subset analysis by flow? And are you doing it, or what are your thoughts on that? If you can share your experience.
Yeah, we were doing T cell subset analysis as part of our PID protocol for children with primary immunodeficiency, as well as for the other patients with immunodeficiency. So we did do in some of our cases, severe cases, and in some patients who did not go into severe disease at upfront and later. And we did find a difference between the, we all know lymphopenia is one of the predictors of severe disease. Less than 800 total lymphocyte count is a predictor. So we did find it that there was a reversal of the CD4 rate ratio in the patients who had severe disease. Though we have now are trying to go into the subset analysis for the central memory and the effector cells if the third wave time, to see whether that has a role, because that will be the post-vaccine situation where the central memory and effector memory cells are the S spike protein reacted CD4 cells. So that would be at a later stage, but we did look at this. And recently there's been a systematic analysis of 61 studies published in the PLOS, where they have clearly found that the routine normal people who don't go into severe, when they come up front and on day 6, if their T cell subsets are normal, they are not likely to go into severe disease, whereas the ones who have reduction in CD4, total reduction in all the T cell subsets, as well as reversal of the CD4 CD8, are the ones who are likely to go into severe COVID. So there are studies, people are planning, and there's a trial going on, planned and approved, to look at this upfront to see whether it's a predictor of severe disease, because there's a lot of data from abroad which is pointing at that when you are doing the CBC, when you're looking at the neutrophil lymphocyte ratio at the same time on the same EDTA blood sample to do a T cell subset analysis and see what it does predict.
Oh, yeah.
But today, as it was being discussed about serosurveillance, though the government of India has approved antibodies only for serosurveillance, not to look at the National Health Mission has also not approved it to look at your immunity post vaccination. But everyone is looking at the IgG neutralizing titer to see whether it's a post vaccine effectiveness. But there are people who don't have it and they have a T cell immunity. So the T cell
Yes.
immunity can be looked at by doing the subset analysis, and it's being standardized, though yet it's not standard of care.
Yeah.
It is being standardized by looking at the memory effector and memory cells, central memory cells, and the effector memory cell ratios.
Yeah, I think that would be a good strategy to actually evaluate, standardize, and
In the
evaluate
future, it may
the.
replace the
Yes,
serosurveillance also.
exactly, exactly. So now we come to the discussion about what would be a natural a flow of thought is about the variants, the COVID variants. So, and what are the implications about COVID variants in India? Because every other day we hear a new variant coming up, and literally we are about to exhaust all the Greek alphabets right now with the naming of these variants. And what is the need for sequence based testing in it? So, Dr. Partho, if you can just throw some light on the variants that are there and about the sequence-based testing.
Yes, thank you, Prabal. And I think Dr. Jyoti had really brought up that good issue, and I hope that the ELI spot test comes into action sooner than later. Now, between the virologists and say the public health domain people, there has been a lot of controversy, debates, etc., as to what does these mutations mean. Now, of course, it is easy to say that all things that is happening is probably because the virus is mutating, or is it that we are looking at the whole database very very closely in real time? So it is possible that in retrospect we will find that for a novel virus, the way mutations are coming up, it could be just a normal progression evolution, nothing happening probably out of the normal, like it had happened with H1N1 swine
Yeah.
flu. Now, still, it is important to see the variations. Most of the variations that are happening are rapid and does not have much of effect on the overall scenario. However, a few of them are definitely that needs to be looked into. So WHO has termed them in the way now they call them as the variants of
Interest.
concern, or the variants of high consequences, or variants of interest. And a new thing has come up called the VUM, that is variants under monitoring. So now, why it is important? Because with that we know that how it spreads, or whether the disease severity is higher, or is it going to be more difficult to control it with therapeutics, or vaccines will work or not, or does it have a social and a public health impact? So, as far as the variant of concerns are concerned, it has been found that yes, their transmissibility is higher. They may be little less effective towards the vaccines or other public health measures, and of course, they have got a looks like they have a increase in virulence. As far as and WHO has two or three organization names them. So the one we follow is the WHO pattern, which goes by the name of Pango, and they name it as alpha, beta, gamma. There's another one also, GISAID, next strain, etc. Now, in India, what we find is the variant of concern that is circulating is the Delta variant, and there is a lineage called AY lineage. Now, unfortunately, what has happened is that we are like in my case, I mean, now still till today, I have tested about 18,000 samples pan India, and we are submitting these, but somehow government is not making these variants or these genomic sequences public. Now, until unless the lay doctor or the people know exactly what kind of variants are working here, how do we prepare? How do we make our I mean, make our capex, or make our prepare this more robust, if we don't know that what actually which are the variants are circulating? Whatever is coming is just by the word of mouth. Now, that is what is very important. And of course, a large number of variants are also under monitoring. And at the end, it just seems to me that mutations and variants are only one part of it, but the triangle of host and the environment and the agent, there are a lot of things apart from the mutations. So mutations are there, vaccines will be there, but the question whether the vaccines needs to be tweaked as it evolves, we may not yet get an answer, because it is like that metro railway station. You stand at one place, the door opens in front of you. You keep on moving to one place, the door also moves. So as of now, it is difficult to say. But yes, these are the variants that are there in India. It is the Delta Plus, or the AY lineage that is likely to be there. Thank you.
So, Dr. Sanjay, if I can ask you your thoughts about, do you see that in the near future, sequencing-based testing would be given to the corporate sector, or it would still remain within the government agencies only?
Yeah, Dr. Prabal, I think if we were to take, say, benchmarks that maybe the UK government is doing, where they have a set percentage of the number of cases that must go in for sequencing. And
Yes.
if if our government was to follow the same, whether it's 5% of all positive samples that need to go in for sequencing, then I think they would need to widen the net and they would need to bring in the private sector to definitely help, because then I think it will not be managed. Currently in Mumbai, we have just one of the municipal centers that is doing sequencing. And as Dr. Partha said, that I think the information is not easily accessible and available.
Yeah.
I spoke to one of the journalists who's following COVID very diligently, basically to look at if every breakthrough infection, every infection that happens post vaccination, a large percentage of them underwent sequencing, and that information was shared with vaccine manufacturers. I think it will go a long way in being able to modify and adapt their strategy on how they need to tweak the vaccine for the local population. I just think it's information that is just sitting there. We know the people who have taken the vaccine. We know how many of them have had breakthrough infections. We need to pick up those samples, get them sequenced, get that data analyzed, share that data with the vaccine manufacturers. It's a win-win situation for everybody. Somehow, I think I'm not clear why this kind of a step is not being taken more aggressively. I think if the government takes that stand like they're doing in the UK, I think there is a lot for us to benefit from that.
Yes, I think there is a lot in this field. I mean, perhaps as we have a better understanding, and in the past 1.5 years, we have really learnt a lot about how COVID is evolving, and we have adapted our laboratory operations, our strategies in a totally different way. And it was great to hear from all of you. I mean, all your thoughts, all your views, experiences about this, what you have seen in the first and second wave, what we have learned, and we are, I would say, we are in a much better pedestal today to face the onslaught of the third wave, if at all it is there. And from this, I would request Nehal or anyone, if there are any questions from the audience, I think we can devote the last 5 7 minutes on question answer session. Any questions that have come in the chat box? I don't see any questions that have been given to us. So perhaps we can touch upon one or two important topics about it. Dr. Partho, if I can request you regarding the various challenges that you see in RT-PCR. Okay, you have been engaged in it quite a bit. So anything that you would want to highlight, share your experiences regarding the false positives and RT-PCR challenges which are there. You're on mute. You're on mute. We can't hear you. Yeah.
Sorry, I have to unmute myself. No, Dr. Prabal, yes. Every day we in the various number of groups that you are, somebody says that, look, I was positive once and then I became negative and then after some day I became positive. So of course, one has to realize one thing, that most of these PCR kits are still not very, very robust scientifically. Yes, they are all working because the technologist has put them on. Most of them are still not maybe IVD. Most of them are still working under emergency authorization. Now, having said that, there are also situations where you would be continuously detecting RNA because there are residual RNAs. People keep on shedding that. I know a number of cases where the person has been positive over almost 46, 47 days, but has been completely asymptomatic. But they have not been allowed to travel anywhere because the RNA is positive. Now, in this case, of course, if the person is asymptomatic, the person is vaccinated, and it continues to shed some residual RNA, it would continuously be coming up as positive. And how do you do then until unless you do a viral culture? Now, as far as the false negatives are concerned, it is very easily we can answer that. Okay, there are pre-analytic issues, or the level of detection is maybe very low. You'll be very surprised that amongst the PCR products that we have, there are products where the level of detection is just about 150, but there are some which are 3,200. It doesn't make a difference when the pandemic is on, because most of them are having a very high viral load, so it will pick up. But as the pandemic is going down and people are vaccinated, herd immunity is increasing, more and more we will have false negatives also. And the moment it is less than 2% or 3% of positivity rate, we go for further pooling. And you see that further reduces your chance of detecting a positive, because already the dilution factor is there. So these are the things that can happen. Yes, false positives with a residual RNA being shed is something which is very, very important, and this is happening even in the Western countries. And some lawsuits have also come where the person actually said that I am vaccinated, I'm constantly positive, and am I infective? I mean, am I do I require to stay under quarantine? So yes, I think this was something which was very interesting scientifically, but a very sad thing for the person who's actually not allowed to move out despite being asymptomatic.
In fact, one thought is regarding when you said about the false positives, about the PCRs. It many times it depends on the, yes, specificity, sensitivity of the kits, and there are many kits which have got single gene target, although it is, I mean, commonly understood that it should be at least 2 to 3 gene target, but there are still many kits available in the market, and some of the rapid tests, the new technologies which are coming up also are targeting single gene. So
Yeah.
possibly that, and that too single S gene, where all
Mhm.
the mutations and everything are likely to be there. So there, I mean, what are your thoughts about this and the S gene dropout
Yeah.
phenomena?
No,
If
it's
there's,
very
yes,
interesting. I mean, whatever you said is very interesting. Now, the thing is this, that suddenly the aim was to make these kits much more specific. That means you wanted a N gene, you wanted a S gene, ORF1, etc., etc. Now, you see, we always said that e gene is a non-specific target, we should not actually use it. But if you see biotechnologically, bioinformatics wise, the e gene, that is the envelope gene which is common for all the coronaviruses, is something which is has got very low stringency. That means if there is a coronavirus, it will come positive. Now, that was a very good screening test actually. E gene was a very good screening test because that could be picked up very easily. Right. Now, the moment you have removed the E gene from that, otherwise, if it had got a very good negative predictive value. There is no E gene, okay, it's not a corona, I mean, it's not COVID-19. E gene is positive, then it could be one of the many corona, and you do a second test, or you multiplex it such. Now, whenever it came to N, or whenever it came to IDRP, ORF, these kits that came up, many of them, as I'm telling you, it's not very perfect as yet. So these are much more stringent, and it is easy to miss if you have only these specific genes of COVID-19. So yes, I think of course, it should not be based on one gene; it should be two gene, and I will still I mean, I'm a proponent of that original E gene, envelope gene, because that had a very good negative predictive value actually.
yes, I think, I mean, you are right in that. I mean, as far as possible, we should avoid single gene and based kits, and if they are especially on S gene, that would remain a challenge today. Okay. So, if I mean, as a last word, perhaps a thought from all of you. What are your thoughts about whether the third wave will at all be there or not? Because that's an eternal question, and maybe it will help me to be ready for the third wave. So let me start off with Dr. Jyoti. Your thoughts? Would there
It
be a
all
third
depends
wave,
on
ma'am?
us. And the crucial time is going to be post Dasera.
Post
We'll come
Dashera
to know
Diwali? Yes,
a
November.
week post Dasera, we'll come to know whether we are getting the third wave or not. I think that's the time.
Dr. Disha, what are your thoughts?
So it's actually a joke that's been doing the rounds that people are saying, "Oh, let me go for a trip quickly. Third wave is coming,"
Yes.
and they're like, "Bro, you're the third wave." So I think that's basically what's happening. Everybody traveling all over the place might just eventually cause a wave again.
So, Dr. Sanjay sir, what are your thoughts? Because with the numbers that are coming up from United States, I mean, it is actually very a concern that how come it is still there, and what can be the timing that we should be ready for?
I think I agree with Dr. Jyoti that it depends on us. I think the way I look at it is, you know, if we lower our guard, we will get hit. I think that's really what happened between the first and the second wave. We lowered our guard.
Exactly.
I feel to a large extent we have become a little more aware, and I think people are still, I think, a little more wary. They haven't dropped their guard perhaps as much as they've done after the first wave, but it remains to be seen. I think vaccinations have played definitely a big role. There are some places in the country where there is still the second wave is still continuing, like we call the fat tail. Kerala, for example, some parts of Maharashtra, we still see the second wave continuing.
Yes.
Kolhapur, for
Yes.
example. So
Kolhapur.
there are still some places where even the second wave is not yet ended. But I think I completely agree with the panel. It depends on us. I think if we keep our guard up, uh, there's a good chance that we could thwart
Yes,
this.
yes. In fact, just 2 days back, I was at Kolhapur, and I mean, despite the number of cases, the guards were down. I mean, it is again the same phenomena that before the third wave, let us, I mean, enjoy the thing. Okay, in fact, there's one question for Dr. Jyoti, ma'am. Uh, there's a question from the audience. How is your hospital preparing for the possible third wave?
Well, our hospital did prepare for the second wave. We became a complete COVID hospital with just 50 beds left for routine patients. And so the second wave, because we were working full capacity and we had worked up the human resources, everything, obviously we can't go beyond the full capacity, so we are ready for the third wave. We have already planned the stocking of the reagents and how we'll go about it as far as the
That,
laboratory is concerned. And the planning is there. Let's see, we are hoping it doesn't happen. And that's what I said, we are just waiting for that 1 week, because nowadays, in North India, they don't go out during Shraddhs. So it's the Shraddh season. It's going to be the Durga Puja time and the Navratra time, and post the Sera that we'll come to know what happens. So we hope it's not
thank you.
there, though we are mentally
Let's hope.
prepared for the third wave. I hope and pray it doesn't happen. That's the most important.
Let's hope that everybody keeps safe and we do not have the similar situation what is happening in US or UK, and we remain safe. People take the
Yeah,
full
US
precautions.
has breakthrough COVID. My
Yes,
son got it a week
yes,
back after two
yes,
doses of Pfizer's from the hospital
yes.
itself. He's an
That is a good.
endocrinologist there. So breakthrough is
It's
there. So none of us are safe. We have to be very,
exactly. We have
Yeah,
to be
extremely.
very careful.
yeah, and I think the virus is not attending these academic sessions, so it
Exactly.
Yes.
is
They don't
so
read books.
they don't read. So it is mutating, and it is not going to attack us. We are going to embrace it, and whatever
Yes.
variant is at that point in time, we'll say that this is the one which is causing all this problem.
and,
The
and and the
virus knows
virus is
the
wise.
Darwin's theory. We do not know the Darwin's theory. That's what is happening.
The virus is very wise. It is making us wiser, and it's posing challenges at every nook and corner. So we must keep our guards on. But anyway, thank you, everyone. It was a great discussion and a learning experience for me also, interacting with all of you. Thank you, everyone on the panel. Thank you all.
Thank you, Dr. Prabal. Thank you
Thank
for a
Thank you,
wonderful
Yes,
session,
Dr.
Dr.
Sanjay,
Prabal, and
over
thank
to
you,
you.
everybody else. Thank you.
Thank you, Dr. Prabal Dev, for moderating this panel discussion, and I thank each and every panelist for joining this panel discussion and sharing your views, opinions, and intelligent guesses for the COVID-19 possible third wave of COVID-19 pandemic. As a token of our appreciation to your contribution, we'd like to present certificate of participations to you. Thank you, Dr. Sanjay Arora, for participating
Thank
in
you.
this panel discussion. Thank you, Dr. Colonel Jyoti Kotwal, for participating in this panel discussion. Thank you, Dr. Partha Roy, for being here. Thank you, Dr. Disha Bhatia, for participating. Thank you once again, and stay safe.
Thank you, everybody. Take care.
Thank you, everyone. Thank you.
Bye-bye.